Hi everyone! This video will guide you through more details about a new biotechnology that can be used to treat ALS. In this video, you will learn about what is a CRISPR-Cas13 based platform for ALS, how can this help your patient, how can this technology help with the treatment of ALS, how does this technology work, how could this technology improve the clinician experience, and how could this technology address a need in the patient experience for ALS? Firstly, what is CRISPR-Cas13? CRISPR-Cas13 is a programmable RNA targeting system derived from bacterial immune defenses. It uses a guide RNA to direct the Cas13 enzyme to specific RNA sequences where it cleaves and degrades the target RNA. Unlike DNA editing tools such as Cas9, Cas13 does not alter DNA. It temporarily silences RNA molecules, making it ideal for blocking disease-causing proteins without permanent genetic changes. How can this help your patient? The CRISPR-Cas13-based platform for C9orf72 linked ALS can potentially be used to remove the unusual parts of your genes that cause ALS symptoms. And it does all these with minimal side effects on other areas of your body and brain. Moving on, how can this help with the treatment of ALS? ALS is caused by a mutated version of C9orf72 gene that normally codes for proteins that control sending and receiving signals between neurons. The CRISPR-Cas13 is able to inhibit the translation of the incorrect C9orf72 gene by directing cleaving of the RNA. This prevents the effects of the incorrect C9orf72 protein from manifesting. And now, let's talk about how this technology works. First, scientists look at cRNAs for RFX-Cas13D using a dual luciferase reporter screen. The cRNA is like a little guide. It looks for the part of your genes that are not supposed to be there, and then it will tell the Cas protein where to cut. The RFX-Cas13D is a Cas protein. It will then goes to where the cRNA directs and cuts the incorrect sequence. The dual luciferase reporter screen is like an interview. It will make sure only the best cRNA and RFX-Cas13D are used. Next, scientists will genetically engineer N2V8. N2V8 is a high-fidelity variant of the Cas13 RNA targeting CRISPR enzyme that helps maintain a strong RNA cleavage and also minimizes off-target cleavage. Essentially, it will make sure that enzymes reaches the correct target and will not accidentally cut unintended strands. Next, scientists will design a guide RNA that is most compatible and effective with the Cas13HF protein and package both into an AAV9, which can effectively target cells in the central nervous system, thus being the best option. This is like ensuring an important package is delivered by a responsible mailman who knows the path there. This package is then inserted into the body through spinal fluid or intravenously, replacing the body's old RNA cleavage with a more effective treatment. This is like replacing a poorly trained supervisor with a new one that is more competent and can catch more mistakes. Now, let's talk about how could this technology improve the clinician experience. The CRISPR-Cas13 targets the root cause of ALS, treating the disease rather than just the symptoms. Its high specificity reduces off-target effects and minimizes side effects. The therapy is compact and can be delivered efficiently to the CNS with fewer treatments. Finally, let's talk about how could this technology address a need in the patient experience for ALS. Unfortunately, ALS currently has no disease-modifying therapies, with treatments limited to a palliative care. Patients with C9orf72 related ALS could benefit from targeted therapies that slow or stop the progression. AAV-delivered Cas13HF enables a one-time treatment, avoiding frequent hospital visits or daily drugs. And that's it. Thank you for listening. This is our works cited page.